Well, hey there, folks. Dr. Klutsky here. So we're going to be getting into Chapter 5 today. And as you look on the syllabus, we are scheduled for an exam at the end of the week. I'm down with that. We have had, earlier I mentioned the possibility of doing a Zoom call review, and I'm thinking I'm going to set that up for Wednesday. Ideally, just because I'm going to set up Wednesday at 11 o'clock, which is our class time. And feel free to Zoom in. I'll record the whole Zoom thing and post that. So, if you got questions, or I'd love to see your shiny faces, or hear what you're thinking about, or listen to stories of bacteria. And we'll put together a test, and I haven't decided if I'm going to put it on Moodle or if I'm just going to give it to you. But let's be honest, it's not going to be that hard, and I'm not going to be that crazy about it, Hey, you picked a good semester to take micro. Hopefully you're learning something. That's really all I want you to do. The assessment, whatever. But I would say that my goal is to give it a time limit of some kind, and so you have to at least have read through the material and have some ideas. Otherwise, you wouldn't have enough time to get all the questions answered. But we'll see how this goes. If you don't know me very well, I got to tell you, I am big on tests and assessment. I'm big on learning. If you're learning stuff, hey, we're doing good. So we'll also have a little reading and review assignment coming up as well as part of the things that we do for every exam. Anyway, today we're going to start talking about Chapter 5, which is bacterial growth, and a little bit of ideas about nutrition, the kinds of things, or the way we classify bacteria based on what they're eating. So this is actually some very cool stuff in terms of how cells divide. And so I thought we would just start with a little whiteboard discussion, and then we'll shift over to some slides. So bacteria undergo what is known as binary fission. And what this means is the cell is growing. It's got its DNA in there, and it continues to grow, and it'll actually start to get a little bit bigger, longer, and DNA is growing. And once it's accumulated enough material, enough nutrients, it can actually go ahead and begin to replicate its DNA. One of the things I always think is very interesting when we start talking about cell division is that cells have to be very careful when they replicate. If they don't have enough energy and enough raw ingredients to carry out DNA replication, they don't go into it, because if you start replicating your DNA and you don't have enough energy or conditions aren't right, you can end up screwing yourself up. You can end up making a progeny that's got some screwed up DNA. I mean, the worst thing that could happen to you is you decide, okay, we're going to go ahead and divide. You start replicating your DNA and you run out of the nucleotide A. It's like, uh-oh. And you got stuff that's halfway replicated, and it would just be a mess. So you never want to replicate unless you've got plenty of energy, plenty of raw ingredients, and everything's ready to go. And so then you go through it, and boom. Because I have to imagine that whenever a cell divides, or it goes into cell division, the cell is a little anxious during DNA replication and during the chromosome separation that everything goes okay. It's a little bit like launching a rocket. You got all these checks. And when you go ahead and launch, that is, when you split that cell off, you're committed. And if you screwed it up, it could be a bad explosion. So your cells, our cells, bacteria cells, they take this rather seriously because genetic defects can have some long-term consequences for the progeny anyway. And it's pretty hard on the individual cell. I mean, it takes some energy to do this. So next, we go through what is known as DNA replication. Now, if you want to get into the real nuts and bolts of this, take genetics or molecular genetics. But the DNA polymerase is going through and separating out the individual strands of the double-stranded DNA and filling in the new strand either direction. And you make a copy of the DNA. All right, so we now have this long shoebox, but now with 2 copies of the chromosome. And at this point, they are going to separate out, and the DNA moves to the 2 poles, so they are being pushed around. And we then get into the fission stage, in which we actually have a cell division event in which a ring, a contractile ring, forms here that is going to begin pinching these 2 cells off. And we then have actual cell division, and we end up with a cell goes off and does its own thing, and another cell is there. Now, I have to say, when you're doing this, at the end of the run, it's kind of hard to tell who's mama and who's baby, because they pretty much are equivalent clones. So this is known as asexual reproduction. And it's asexual, meaning that rather than mixing the genomes of 2 separate organisms, we just have a cell that is dividing and splits apart. Now, your cells are doing that all the time. I mean, this is part of what you're doing right now, is you have cells that are dividing, and they are, in different tissues and things, but they are essentially clonal. They're all you. Now, they're not going to separate out from you and begin developing into a complex organism and walking around, which would be pretty darn cool, but no, that's not how we do. They are simply just populating up your tissues. Now, with bacteria, and actually there are a couple other organisms, yeast do this and fungi do this, the cell can replicate its DNA and split off another cell, and that other cell now can go on to become a full organism. But sexual reproduction is what eukaryotes are doing, where we have gametes, an egg and a sperm, that have just a single set of chromosomes. They fuse to produce what's called the zygote, and then that guy begins dividing and begins turning into a baby. So that's sexual reproduction. And sexual reproduction has the advantage of bringing in different DNA sequences, one from mom, one from dad. You also have the possibility of what's called recombination, and it just gives you a lot more variability. So you end up with within an individual species like Homo sapien, all these different varieties and all the things we are. With bacteria, if you know one E. coli, you probably know pretty much all of them. There's not a lot of difference in these clonal organisms. So, let's go to the screen here for a second, and we'll look at a couple of figures here, just because. I kind of like writing, but some of the stuff gets more complicated, and it's easier to look at a picture. So here's the thing, while I'm using PowerPoint and not writing on the board, that don't mean you don't have to write. You should be writing stuff. Let's crank this guy up. Bada bing. And fine. I got emails. I don't care. I'm going to kill some more lights. Nah, it's coming up. All right. So, we got yeast. They are carrying out something called budding. The main difference here, rather than having a box that spreads and then gets clipped in the middle, budding, you end up shoving some DNA over to a little corner and you pinch a little blob off. And so you really can tell that this is the mama and that's the baby, and then there's bud scars. And the individual guys then go on and carry out their own DNA replication and budding as well. There are organisms like different kinds of molds and fungi. These are eukaryotes, by the way. So are yeast, so you know. They can divide through fragmentation, where they actually just are breaking apart. Now, they have to have, rather than undergoing a specific DNA replication to produce new organism, it's a little bit like taking a transplant or taking a cutting from a plant. So they just break apart, and then these new pieces, they can go on and begin growing up. And so, I mean, it is essentially exactly like taking a cutting off of an ivy, putting it into a little bit of water, letting it root. It's cloning. So these are all examples of asexual reproduction. The only difference is with bacteria, they are doing binary fission where each daughter is, or each split is basically the same size. With yeast, the new guy is a little tiny bud, and then you got the big mama ship. And then with fragmentation, you just start busting them up. They're not really undergoing replication as much as they just kind of break apart and spread around. Now, this is not A&P, but, you know, hey, this is some very cool stuff, and that is thinking about the eukaryotic cell division cycle. And yeah, you probably have had this in A&P, I hope, or in different places, but there's some very cool things to think about. And if we think about the cell cycle in this sort of notion of the criticality at these different steps, there's some very interesting things to think about in that there are spots where the system will do a check. It'll do a hold. Essentially, it's called a stop. And it is a way for the system to kind of do an assessment. Did DNA replication occur properly? Or, I mean, let's just go through this here very quickly. Look, I'm a eukaryotic cell biologist. I find the cell cycle to be really cool. This is part of cancer biology. It's just a very neat area to look at. And you might think that, okay, you're talking about the cell cycle, that's just mitosis. But here is mitosis. Mitosis is just one teeny little spot in this grand cycle. And so this is looking at hours, 15 hours. Your average animal cell probably does about this, maybe 24 hours. So we've got the initial phase, the G1 phase. This is where you begin to accumulate all the material. You have the different kinds of metabolism going on to generate the raw ingredients. And you can then go into S phase, and that is DNA synthesis phase. Now, here's one of the things that's kind of fun looking at eukaryotes that prokaryotes just don't do, is that you can proceed through the G1 and stop here and go into what's called the G0 state. G0 means you're not doing anything. You pretty much are, kind of off the merry-go-round, and you're not engaging in cell division anymore. You're just sort of like, we're going to wait here. We're going to just hang. Now, some cells, like neurons, are pretty much in a G0 spot. They're not going to go back into DNA replication very well. They don't divide. Terminally differentiated cells. But then you can go into the S phase. You can produce DNA. And now the cell has gone from having its full set of chromosomes, one from mom and one from dad, to having two full sets of chromosomes, because it's about to split. Some cells actually will hang here and take advantage of the fact that they've got more DNA and more capacity to generate more proteins and things. So there are some cells that are actually hanging at this spot. So then we go into the G2 phase. That's the next phase in which the DNA has been replicated. We start to replicate different organelles. The mitochondria can kind of split up and divvy themselves up. And then finally, we go into mitosis. And mitosis is the actual condensation of the DNA, the chromosomes form, it's pulled to the two sides of the cell, and then you get the cell division, the contractile ring, pulling the two cells apart. So, because eukaryotic cells are just more complicated, they got a lot more DNA, they tend to be a little bit more persnickety about how they run around this cycle. But bacteria, they're able to cut a cell division very fast. And that's another kind of contrast we can think about, is that the eukaryotic cell systems, our cells, generally we say they replicate about every 24 hours, 18 to 24 hours, whatever. Not all that fast. There are some eukaryotic cells that are very simple eukaryotic organisms, like yeast. They replicate pretty fast. But complex organisms, they take a while. And it just depends on how much DNA you have, because there's a limit how fast you can replicate DNA, and just how complicated a cell system you are. So the cell division cycle in eukaryotes is really broad. Now in prokaryotes, these puppies are hooking it. Bacteria divide, well, every 15 minutes anyway. A rapidly dividing bacteria is absolutely moving out. Now, one other comparison thing just to throw in the slide, we talked about asexual reproduction, in which we're essentially dividing cells and you're making clonal lines. And this is going to be really, this is what bacteria pretty much do all the time. But also, this is what your cells are doing internally, right? They're just dividing. I mean, we actually, it is said that you have renewed every single cell in your body like every 5 years. The long bones take a while. It's not entirely true because your brain's not really reproducing the same way. But in terms of your gut, you shed gut cells every day, and you have to make new ones. Lung cells, all the time. You're coughing them up. So they're all in skin cells. As they slough off, you're making new cells, and these are clonal. They are undergoing pretty much asexual reproduction. I mean, we don't really call it that because it's not independent living creatures. It's part of my skin. On the other hand, sexual reproduction is where we have a mommy and a daddy. Well, what are we trying to show here? We have a mom, yay, and a dad. And the mom produces one gamete, the dad produces the other gamete. And this has gotten an advantage because you do get a greater degree of genetic variety. I mean, if that's something you're looking for, sexual reproduction is the way to go at it. Okay, now, this is another really interesting point. When we think about the growth curve of a bacterial infection or a bacterial culture, and this is based primarily in a nutrient media culture situation. But we can think of this in a lot of different ways. And to be perfectly honest, this mimics the viral load going on in the COVID-19 infection. In that, you start out with a very low number of bacteria. This is known as the lag phase. Now, what's happening here is the bacteria are sort of adapting to the media. They're figuring out how they're going to start growing. This is what's happening when you inoculate a culture or inoculate a dude. You get a bacterial infection, it's going to be a few bacteria. I mean, think about this, a cut on your finger. You may only have, 3 bacteria getting in that wound. A puncture wound, it may not be very many bacteria. I mean, depending on what it was you got punctured by, it could be nasty, it could be several thousand. But regardless, you have just a few. And now, it takes a while, but after, about 30, 40 minutes or so, they're going to begin to go into the exponential growth phase. Check this out. We go from one bacterium, more or less, into a bunch. It is a logarithmic type of exponential growth. So you're growing at a rate of 2 to the n power. N is the number of generations, and it's 2 because you're splitting. Now, this can't continue forever because there's a lack of space, a lack of nutrients. Things are going to slow up. In a bacteria culture, in the lab, they get so thick, they're touching up against each other, they start to run out of nutrients a bit, and they slow up. And that is what's known as the stationary phase. Now, the stationary phase is where you're going to have the most serious medical conditions. So the lag phase is where you have the initial infection. The exponential growth rate is probably not initially going to exhibit symptoms. But once you start getting to be a high enough level of infection, bam, you're going to start having different symptoms, and you're going to know, okay, we got an infection starting to roll here. And at the stationary level, we have kind of reached the carrying capacity of the media. The cells are continuing to divide to maintain that number, but they're not dividing at nearly the same rate. They are mostly just hanging out. But there's so many guys there, so many bacteria, that they are generating toxic metabolites. I mean, they're all peeing in the same pool. And they have to kind of adjust to where the number of births equals the number of deaths. So the bacteria dying, but also bacteria being reproduced. And you have this stationary phase. Now, after a time, the environment becomes so limiting. That is, you have eaten everything there is to eat. There is so much pee in the water, well, bacterial pee, that the death rate begins to actually get higher than the birth rate, and we start to go down. And so, at this point in the medical setting, you're going to see an improvement, but not all the bacteria are dead. And so, depending, you can start back up and go back up again. All right, now just a little point. If you've got a really raging infection and you give antibiotics, there could be a fairly serious effect of killing that many bacteria. I mean, this is what toxic shock syndrome is, in which you've got a lot of bacterial material in the cell wall, the peptidoglycan. As it gets released in the body, it causes a dramatic inflammation effect, and that itself can be toxic. So it's not that the antibiotics aren't going to work, it's that you're too late. That the antibiotics are certainly going to work. It's just that the number of bacteria is so high that you're going to have a really rough time. You're not necessarily going to die, but it's not going to be as easy as if you were knocking it back, say, about here. Now, with COVID-19, it's actually an interesting kind of thing happening, the same thing, because this kind of growth rate is what organisms do. I mean, the human population does this as well. You have the initial inoculation, that's the lag phase. You get enough individuals now that start really replicating and off to the races. And you then end up at the stationary phase. Now, probably we were starting to get to the stationary phase and able to come back down in end of April, 1st of May, and if we had stayed locked down, sadly, for another 4 weeks, we could have very likely brought the viral load in the population down low enough that we would have another lag phase and we could actually coast for a while. But that didn't happen. Anyway, so this kind of growth curve is an interesting way to look at life in general. Now, in terms of bacterial infection, it's also a really cool thing to think about. All right. So now, the next step, and we're going to just touch on this and then stop here. Thinking about the kinds of things that the bacteria can consume in order to generate the nutrients that it needs to actually do all this growth. So we've got a couple of sources of energy, and you can either be a phototroph, which means you are a light eater, or you can be a chemotroph, which means that you are getting energy by breaking chemical bonds. Now, let's just face it, you and I, as much as we like the sun, can't extract energy from photons. Who can? Raise your hands. No, plants can. Photosynthetic plants are able to actually harvest the energy in red light and use that to drive the chemical reactions. They basically are popping an electron into a higher energy state and using that electron's energy now to extract the energy to make ATP. And they're able to actually use sunlight as an energy source. Now, another thing that all organisms need is a source of electrons. And for us, and for for us, we are extracting the electrons from the same thing that we're extracting the energy from. Now, in a plant, they get energy from the sun. They extract electrons, because you need electrons for making bonds. They extract electrons from water. So that would be a lithotroph, where you're using an inorganic or non-carbon containing molecule as the source of electrons. For you and I, we are organotrophs, where we use organic molecules. Now, another component that's required for living is your source of carbon. Now, if you think about us again, we get our energy, our electrons, and our carbon all from the same Twinkie. But not all organisms work that way. You can, in fact, get sources of carbon from different spots. Now, an autotroph is able to extract carbon from carbon dioxide, a fairly inert molecule. A heterotroph is only able to extract carbon from what we would think of as edible materials, from organic chemicals. Things like, a Twinkie, a Big Mac, whatever. So, we as humans, and all animals, we're heterotrophs, and we're organotrophs, and we're chemotrophs, and all of our stuff comes from that stuff that we're eating. Now, from just a different perspective, if we think about a plant, their energy, the driving energy is coming from the sun. They are using water as their source of electrons, and they bring carbon in, not from eating. I mean, there are, like Venus flytraps that eat bugs, but that's kind of rare. Your basic corn plant is not carnivorous. They're getting carbon from the atmosphere. So plants provide an example of a whole different way of operating than we do. Bacteria can run the whole gamut. They can do all kinds of different things. We can have guys that use light. We can have guys that are using organic or inorganic materials. And we then have guys that are using organic materials or carbon dioxide. And we have these different names. So a photo-organo-heterotroph, a photo-organo-autotroph, a photolitho-heterotroph, a photolitho-autotroph, a chemo-organo-heterotroph. Let's just think about what we are. We use chemical compounds. We use organic molecules as our source, and we are using organic chemicals for our carbon. So we are chemo-organo-heterotrophs. And then you've got some organisms that are chemolitho excuse me, autotrophs. All right. So what I'd like for you to do is write down these terms: phototroph, chemotroph, lithotroph, organotroph, autotroph, heterotroph, source of energy, source of electrons, source of carbon, and be able to combine them. So if we can ask you, what you might classify an organism that does certain things, it eats in certain ways. We'll stop here for now and continue on Wednesday. We'll do a Zoom Wednesday at 11. I'll figure out how to send the link out to you and all that. If you can make it, good. If you can't, hey, I'm around, and pretty much guaranteed I'm around from 10 to 3 just about every day. That's like pretty much guaranteed. And I'm in and out, but either hit me up with an email or just come on by. You're welcome to come on in and hang with a lecture, whatever, okay? So that's me. I'm out of here. Let's go to a hot spring bacterial vent.